Iago is research manager of Microalgae Biotechnology team at Wageningen University, and he is passionate about science, answering practical questions, finding solutions and opportunities in the field of microalgae biotechnology. Okay.
Thank you very much, Marie-Christine. Good morning, everyone. I'm very happy to be here. Thank you for being present and for your interest in our project. And following on Massimo's introduction, there is one big issue with microalgae, that it's costs, of course, and resource efficiency. And that is the title of my talk today. That's the result of a group or effort that I'm going to show you. But we wanted to improve. It's not moving. Yes, it is. So very quickly, who we are, and bear with me, I have a challenge. We are 6 partners in this, in our work package, focused on the issues of improving production, and I have the challenge to talk about it in 20 minutes. So I will be quick, I will be brief, and if you're curious, I want to incite your curiosity to ask us questions afterwards, so bear with me, that's the results will be quite summarized. We were 2 universities in the Netherlands, Wageningen University, where I'm from, University of Twente, 2 research institutes, Norse and Green Collab from Portugal, and 2 industrial partners, algae producers, also from Portugal, all microalgae and Acton. Our goals involved cost reduction, improving production, increasing sustainability, so the sustainability performance of our the production, and of course a very noble and task that was supplying biomass for our partners working on product development, extraction, as you hear after me. Well, the introduction, it's not really necessary, just some pictures of the beautiful creatures that we work with. Massimo already explained why microalgae are relevant. Some people call them primitive plants. If there are other biologists in the room, we usually cringe at that definition, but it does the trick to communicate. We can find them in different environments, from salt to freshwater. What they have in common, because they are very different creatures from each other, they usually do photosynthesis, which is a bit ironic because we are going to talk about also heterotrophic production. That means algae growing in the dark. And also, we clump them together with the seaweeds. Often. We can do a lot of things with them. That's why we are here. That's why we are here in an innovation action. And this is a brief list of possibilities using microalgae. So we can use the whole biomass as a source of nutrient, as a food supplement, feed, aqua feed. But what we are interested in this case is proteins, but they also have sugars, oils, special oils, polyunsaturated fatty acid, pigments, and so on. These are the 6 chapters. I told you I had a difficult task today. So I divided that in 6 chapters with some hopefully interesting titles. But I go to the first one. If we need to improve production, one of the things we did was developing novel strains that have better industrial properties. We wanted to explore nature's potential, so we worked with non-GM techniques. Of course, in Europe, that is still a discussion. It's not allowed. And here I have some pictures of where it starts in the lab, in a petri dish with agar, gelatin, nutrients, and they are growing. I don't know if with the spotlight if you can see, but they go from very green on the. Do I have a laser here? No. No. From very green to less green. And the question I think that arises from it is, why do we need less green algae if they're naturally green? Well, the color is not really easy to work with when you're formulating food stuff with them. You see on the left, the actual green color of the biomass, and to the right, different variants, mutants. Like we all are mutants. Everyone here has different mutations, all of us. So, do microalgae. We use random mutagenesis, a lot of selection at lab scale, but the final results were 6 novel strains with improved capacities, 3 pale green. You can see them on the top. They are lighter than the original one, the wild type, the so-called wild type. 2 yellow and 1 white strains. Some of them had higher growth and a little bit less proteins. Others had lower growth, but higher protein content. That means a higher protein productivity at the end of the period of production. Both of them, all of them showed improved glutamic acid content in comparison to the standard sources for human nutrition, and that indicates a stronger umami savory flavor. So that has implications for its use in food formulations. Chapter 2 said sun and sugar. We're not there yet in the darkness, but we are working on under mixotrophy. You know, autotrophy is when the organisms can produce their own food, like plants, algae, with photosynthesis. And heterotrophy is when they have to take up their food. They cannot produce. Mixotrophy is when it happens simultaneously or close to simultaneously. So we let algae grow under sunlight and sugar sources. For this, we work with a very exciting organism called Galdieria sulfuraria. The name gives a hint. It was isolated from volcanic hot springs. It's a very lovely environment. That means there is a lower risk of contamination, simply because no other organism wants to grow where Galdieria grows. That's why it is called a polyextremophile. Sometimes algae can grow at higher temperatures, but the rest of the conditions are, let's say, less mesophile, so not extreme. But this one likes it rough, you know. Life is difficult, but not for Galdieria. There is another reason, of course, why we work with Galdieria. It has 60-65% protein, and considering the standards of the FAO, you can find in Galdieria a higher content of methionine and cysteine, 2 essential amino acids. Our approach, this is summarizing the life of my colleague, 4 years of the life of my colleague. I will tell you his name, sorry Pedro, but that was the time that it was available. We started with a scale down. A scale down is when you already know how to scale up, how to produce something in a higher scale, and you scale it down to improve it further. So you know somehow what you're going to do. So the first step was to work on simulations and some pilot conditions at lab scale, bring it down, feed the models, design and simulate the supply of glucose, that was the source of sugar we were using, and then go back to the scale up, which we did in an algae park pilot facilities in the Netherlands. So here we have 2 of my colleagues and someone that I had to hide for privacy reasons, with Galdieria growing in our greenhouses in the Netherlands. Yes, it is sunny in the Netherlands once in a while, but we have state-of-the-art lamps for the very dark days, but these runs were done in the summer when actually it gets pretty unbearable to be inside the greenhouse. More pictures of the different runs. We did autotrophic, mixotrophic, heterotrophic runs. Here's my colleague Pedro. Please raise your hand. Watch out for your coffee. If you have specific questions like how many grams per liter can you produce? Well, actually I remember, but I'm not going to say it now, just to keep you curious. You can ask him. But the summary of 4 years of his life is that Galdieria can use sugar efficiently. It opens up the possibilities to use other waste sources. We achieved a stable production of this polyextremophile. There are very few records, at least officially in public, about growing this strain at this scale under the conditions that we did. So that was a very good result for us, a fulfilling result for us to achieve. And the productivity is within the range or a little bit above, but that is a question for Pedro later. He also has a poster, so please contact him. Now I want to talk about microalgae and insects, the unlikely synergy between the 2 of them. We here used waste from insect production, so insect frass, as a nutrient source for microalgae. Basically what we did, we tested different waste sources from industrial, municipal waste. From this selection, insect frost was identified as the most suitable, both due to the amount that, of its production per year and the nutritional value they have, so the nutrients they can supply microalgae with. Then we had to optimize those trials at lab scale. We used Chlorella vulgaris, a very industrially exploited strain, using it, and of course it finished with the data analysis, comparison, and studying the composition of the biomass. Being very brief here, it's again my colleague Dorinde Kleinegris. Raise your hand if you want to know more. She's your person. These graphs here, being very simple. The left one, top left, you see there's no difference. Everything's overlapping. That means that the controls and all the different treatment that we tested show the same growth. And here, on the right, you see that the protein content has some variation, but that is not considered statistically significant. So when you compare controls with the medium containing insect frost. Then now we go to the darkness. I promised you some lack of sunlight. Microalgae in the dark, heterotrophic growth. Microalgae can also, most of them, I don't know if most, but a lot of them can grow also, can be fermented like other microorganisms. And it has some advantages. Often, when they grow in the dark, they don't have to be as green as when they grow under autotrophic conditions. So they need the pigments to capture light and convert light into the food that they need to live. So when they're heterotrophic, it changes a little bit. So we tested different strains at lab scale to develop the developing trials for developing and improving the production protocols. That included also the novel species from chapter 1. They were also sent to the partner, all microalgae, to be produced at lab and lab pilot scale. In this biomass, both wild types and novel strains were sent to partners for trials. Here, briefly, Elena, say hi. If you need to know more or if you need to buy some biomass, you can ask Elena as well. Our microalgae can supply you with that. On the top, we see some of the novel strains we developed, the mutants. Here, the lab trial, just to give you a taste of what it looks like in the small scale. That's what it is, the vessels, covered because they have to be in the dark. And bigger vessels for pilot industrial scale production. Now, the CO2 paradox. I don't have a picture of my colleagues from the University of Twente. They are not here today, but if you have questions, you can ask me. I can redirect you to them. I call it a paradox because we hear a lot of CO2. It's at very high concentrations in the atmosphere, but there in the atmosphere, luckily, down here, it's 0.00 something. And that's not enough to optimize your microalgae production. What people have to do is to buy CO2 in bottles, pressurized or liquid, in liquid form, and that represents also a cost and an environmental burden, you know. What we wanted to do here, our colleagues from Twente designed, built, and tested a pilot DAC, direct air capture unit that was placed in all microalgae. You see there in the installation, which was today's 13, right, of September. It was literally 1 year ago, right, Elena, that we were there. This is the pilot already installed. It's a vertically stacked container for you to have an idea of the size. This is us, annoying the people that install, you know, after people install something, then the visitors come and want to see things. And this is all of us doing the technical visit. You can see some reactors around it, but you'll see better pictures in a bit. But this was September last year. How the process works. I'm not a chemical engineer, so it's not oversimplified for all of you. It's oversimplified for myself as well. Basically, there is a vent, atmospheric air is being vented in. The CO2 is adsorbed by the sorbent, by the particles, and lean air is sent out without that CO2. That's 0.00 something. With time, you saturate the sorbents, and then by playing with heat and cooling under vacuum conditions, the sorbents release the CO2, and that gives us a stream of CO2-rich gas, which can be 95% pure, so high quality CO2. And if you have, if there are investors in here, yes, we could use the CO2 for other stuff as well, not only for microalgae, keep that in mind. But the pictures on the left show us the green tubes around the unit, and that it's not only for our picture purposes or PR. Those tubes were running on the CO2 supplied by that unit. This data was gathered. It's included in our reports. This was from September, December 2022. We produced about 150 kilos of CO2, like I said, a purity of 95%. And one remark is that the weather conditions were not very optimum. So the relative humidity was above 75%, and for some reason that I cannot explain you today, this is not good for the process. So then take my word for that. Here below there are some graphs and numbers, not for you to read now, but that means that the logging of the temperature, performance, efficiency, costs, and where the energy is going, they were gathered. So these numbers are available for sustainability studies and assessment of the technology. Then we go to chapter 6. I hope I'm not too okay on time? Okay. Have enough? So I'll talk slowly. 6 is reducing consumption of a energy consumption, resource consumption, in this case mainly energy, of a microalgae production facility. Here you see another person that can help you improve your process, or maybe contact you with buying some more biomass as well. That's Alexandre, say hello. This was the Necton. They live in this horrible place called Algarve in the south of Portugal, you know, bad weather, and they have a tough life in there. What we did in this chapter was, well, is design and installing novel reactors, not novel but new reactors, using state-of-the-art knowledge. They were made of glass. That has some implications for the sustainability aspect of the process as well. Glass is more expensive, but lasts longer as well, and gives you our, let's say, cleaner and more controllable process. The PVs were already installed in Necton, but they were optimized, connected to the control systems, and that was done with the goal of achieving an off-the-grid reactor. Come with me. Let's see if I can explain this clearly. We're talking about 27 cubic meters of tubular reactors. There's, we have the drawings first. Remember that Necton is placed in a very beautiful location, a natural park, so it's not easy to have permits and to have this installation. But here they are, inoculated, beautiful, green. And we did a few trials. So the first trial was, and here I have to explain you a little bit. The main part of the process of producing microalgae, the main part that consumes electricity, is often the pumps. Algae are in the liquid, they are in suspension, and you have to keep pumping them so that they recirculate around the tubes, right? The standard practice is to have the pumps 24 hours on, you know, even at night when they are not growing. If they don't have sugar in this case, they are growing on sunlight and CO2 only. So the first test we did was what happens if you turn off the pumps. The first reaction of people working with microalgae is panic. I mean, they're going to settle down, they're going to die, they're going to collapse, and we're going to die, all of us. That's the first reaction. But what we showed is that you can actually turn off the pump at night. They will settle down, and apparently they only fall asleep, you know. And they wake up in the morning and they were fine. So we compared two conditions, completely off, and turned on 5 minutes every hour, check the effect, swapped between reactors, and it was a very positive result, we believed. The other trials were already in duplicate with the new systems, and we wanted to run them completely off the grid. One of the reasons why the pumps had to be off at night is that if you want to run it off the grid, either you put a battery or you turn off things at night. And since we found out that we could turn off at night, it was possible to compare them. And this is another picture. We had an on the grid and off the grid reactor, one next to the other, swapping them for controls. You know, we're all scientists and we want things well controlled. And comparing the growth, we see very little difference between the two modes when we compare trial 2 and 3. Another thing we did was to solve another issue, the issue of contamination. We had already tackled the subject with Galdiaria. So with contamination, you have a few options. One, to be a clean freak and keep everything very clean, you know, quality control to avoid contaminants coming. You might ask, what are contaminants? Many things could contaminate your culture, but normally bacteria, they are not detrimental, they don't collapse your culture, but the grazers do. The grazers are animals that, although are microscopic, they are much bigger than the algae, and they literally eat them. And they go grazing, grazing, grazing. The culture on the left, it's green, healthy. The one on the right, it's collapsed. They can collapse your cultivation in a matter of hours or days, depending on how bad they are. Or good, if you look from their perspective, you know. You can, of course, be very clean, like I said. You can use extremophile, like Galdiaria, that grows under conditions that do not allow other creatures to grow. But you can also, you have to find other solutions. Sometimes they show in your culture, and if you can do something, you might save your production. So that's what we did, checking different nitrogen sources and different strategies. We came to ammonia as a replacement for nitrate. Normally people use nitrate. And in this picture, actually, you see on the right the control with nitrate and the left one with ammonia, and the contaminants that we identified in those trials. They didn't like ammonia, so that is one possibility. Microalgae can withstand the change in nitrogen source, but some of the contaminants might not, and that was also a very interesting result. And here I'd like to summarize what I told you here today in all 6 chapters. We developed here to achieve the overarching goal of reducing costs and improving production and efficiency, both from the production and sustainability aspect. We developed novel strains, mutants with improved industrial properties. We showed that insect frosts can be a source of nutrients for microalgae. We scaled up a polyextremophile under industrially relevant conditions. We found strategies to reduce contamination. We tested and we developed, built and tested a CO2 direct air capture unit on the grounds of a production facility, and we ran reactors off the grid. With this, I love this picture here because it's from 2020. I don't know if anybody remember those years. Something happened. And this is to show my appreciation to all my colleagues. Some of them, they are here, not all of them. But this was really a group effort and a lovely group of people to work with. And it's a pity that our work is done. And we still have one meeting in November. I will chase all of you. And of course, thank you to all the partners of ProFuture, to Massimo and Fabio for having us, and Marie-Christine and her team for coordinating. And if anybody has questions, I'm happy to answer or to tell you I don't know it, but I will look it up for you. Thank you very much.
Are there any questions for Iago? No? Okay. Maybe if you could shortly introduce you when you ask a question, that would be nice.
So I'm Johan Robbins, I'm from Ilvo in Flanders. In one of your first steps, you look to alternative species, But from the regulatory point of view, it's actually an extra burden you have.
Sorry, I didn't.
You're looking for new species. You want to use them in food and feed.
Yeah.
But the regulatory, it's a new species, so you have to do actually a lot of regulatory work before you can ever use it.
Thank you for the question. Again, it goes into that warning I gave in the beginning that I was not very clear and quite on purpose. It's a tip to get questions. With the novel strains we developed, they were all from Chlorella vulgaris, is a strain that it's already accepted and commercialized. And since they are not GM, they are just variants of it.
Yeah. Galdiera?
Galdiera, yes, it is still novel. The last information we have is that if somebody knows that, please help me, but a request is being prepared by some actor in the field, right? For Galdiera, yes. But you are right. Every time you work with a new strain, you have to consider that burden of doing that. I think Javier had a question, I think.
Thank you. Javier Berterreche from Bridge to Food. I have a personal doubt. When you say, for example, in the case of Galdiera, all the extremophiles are better to pick because there is less contamination. I understand this when you pick them, but what happens after that? How you avoid contamination?
The perfect question. When we grow them at a lab or industrial conditions, we try to mimic the conditions they come from. Of course, not the sulfuric part of the hot springs, but the hotness and the acidity. So by keeping the pH 0, it can grow from pH 0 to 4. That is a quite unbearable pH for most microorganisms. So far as my knowledge go, only a few fungi can actually contaminate it potentially, but most bacteria cannot even, unless it comes from a hot spring. And the chances that you're going to find organisms that can grow in hot springs in Wageningen in the Netherlands, it's very small. So we keep the temperature very high and the pH very low, and that keeps the contaminants.
So the second part of my question, I understand this. Do you think you can scale this up at an industrial level?
Yes, and that's what we showed in this in the trials we did. The systems that we worked with, they are commercial systems. Those tubes that you see, they are from the company LGEM in the Netherlands. So they are scalable. You have to take care. The main challenge is if you already have established microalgae production, having your medium with a pH 0 might be a challenge for you, because at this pH could even be corrosive to certain parts of it. So yes, if you already have a production facility, it might be, you have to consider if the investment be worth it, because you have to prepare everything to withstand 50 degrees and a very low pH. But if you can tackle those things technically or investment-wise, there's nothing forbidden you. Yeah. And by the way, there are some information that some industrial actors here and there in different countries are already scaling this up. But of course, if it's an industrial project.
Yeah, I have a question. I am Estelle Coillier from Nantes University in France. And you have described several strains with different colors because you've cultivated them in heterotrophic conditions, for example. And is there any impact on the composition of the microalgae? Because of the photobioreactors in autotrophic conditions, they use autotrophic will lead to the production of specific interesting compounds that could not be produced in heterotrophic conditions. So do you have any data on this and nutritional interest?
Well, thank you for the question. Again, I will give an answer and ask my colleagues to complement if they see something missing. There will be some changes in some components, but the macro components, we focus heavily on protein content, right? So the pigments, of course, they tend to go down Although there are reports of strains that remain green under dark conditions, heterotrophic conditions. So the pigments will disappear, but that's exactly what people want. But as far as I'm concerned, there are some minor changes, but no major change in the composition, right? Like that impactful.
There can be, but we can also. Okay. There can be significant changes, and we want generally to lower the chlorophyll content because the taste is very pronounced. But we can also change the medium conditions or try to optimize a given reactor conditions and parameters to stimulate the production of specific compounds. So for now, we have optimized to increase the protein content and growth, but we can also try to fine-tune the medium to produce a specific lipid or increase the content in some mineral or some specific nutrient that we want to enrich our biomass in. Does that answer?
Thank you very much. Angelos Harlaftis, Epafos Advisors. Have you investigated, researched these microalgae systems? Can they exist in minus 300 Celsius or pressures which are not on the earth and environment? Because as I understood, they need darkness, they need sugar, and they need CO2. So only CO2. So they produce oxygen from the CO2. Have you investigated such a thing?
Are you considering taking microalgae to the space or
To Mars.
to Mars? Yes. Well, not in the scope of this project, unfortunately, because it would be very cool. But I happen to have a colleague in Wageningen working together with the European Space Agents. They are investigating the possibility. But minus 300 Kelvin, you said?
The minus depends also from the pressure of the environment.
Well, that I certainly don't know if we went that extreme, if anybody has a record, but at least to provide, like to take up the CO2 from like the International Space Station and to provide a source of nutrients for astronauts, for example. This work people have been trying and studying. Yeah. If you really want to know more, I can introduce you to a colleague of mine, but that's outside the scope of this project. And if you know anybody working on it, please call me. I would love to join such a project. Huh? Go to Mars, indeed. Anybody else? Going once? Going twice?
Then thank you very much.